this section is a little bit of extra information on human papillomavirus and basically how it copies itself and how it gets into and out of the nucleus. Yeah, the truth will surprise you, I would imagine. So, the star of this part of the show is the L2 protein. You remember L1 made the little coat around the outside, as shown up here. And the L2 protein is that little thing that's like dangling on the inside and doing a whole bunch of stuff. So here's a little map of the L2 protein. And so it's got a furin cleavage site. That's where it actually gets activated. And it'll poke this little part up out of the capsid so it can get cleaved, like a little, me please, raising its hand. Once that's activated, then more of the L2 will stick out of the capsid. It's just like a signal that gets it pulled out, basically. After that, we've got a transmembrane domain, which you can also think of as a membrane anchoring domain. Then you've got a chromatin binding region, because this virus is going to attach itself to your DNA, or rather the DNA and protein that we together call chromatin. And this is the region that it uses to do that. It'll also interact with like a sorting nexin 17, so that's what this SNX 17 is. And down here at the end, it's got a membrane destabilization peptide. And so think of this as like a little pin at a child's birthday party that it can use to pop the balloons, right at the end. So that's what the virus is carrying with it. This is our little Swiss Army knife that the L2 protein has. Now, let's see how they use that. There we go. Okay. So, here is the first step. The virus comes in, and this is kind of the first little dirty trick that L2 does. It's going to be pulled in by attaching to all those proteins. Remember the integrins and some of these other proteins. And that's going to get it pulled into an early endosome. And you know what happens when it gets to the endosome. We're going to start running hydrogen, H plus ions into that endosome. We'll pump them in with the vacuolar ATPase, just like we always do. And once that is done, the pH is going to start to go down, 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 just like it does for coronavirus or any of these other viruses that are sensing acid to know that they are inside the cell. However, in this case, our virus is not going to play the sort of quick escape card. It's no Houdini papillomavirus. Remember, it's very lazy, and so papilloma is going to take it slow. Papilloma sticks out some of these L2s. So the collapsed L2s are shown here as little eggs, and these long sort of, chiclet-looking things, those are L2s that are sticking out of the capsid. They're pulled more or less out. They'll actually stick through the membrane, because remember there was a transmembrane region, and they'll have a little bit that sticks out on the other side of the transmembrane region, and that is going to bind to the sorting nexins and a bunch of other things, which are going to get this entire vesicle. So instead of being dragged toward the lysosome and getting more and more acidic, it's going to be dragged and diverted. We're going to divert it back over here and merge it into a thing called the trans-Golgi network, which is kind of like a waiting zone for things that will eventually maybe go up through the Golgi. And so, rather than jump straight away into the lysosome and have to deal with all that acid, the virus instead just decides to play the long game. So, like it says, this buys a little more time for L2 to plan its escape. All right, so what's it doing with this time? It's hanging out, because if it's in a basal cell, that basal cell is going to divide. Now the cell layers above it, if I remember correctly, either don't divide or do not divide often. It's the basal cell that divides, and then it pushes up copies of itself that become the epithelium. And eventually it will get sloughed off, but that's okay, because the basal cell keeps reproducing, keeps making more little cells to push that layer up. And so as it's doing that, during the process of cell division, the nucleus is going to be dissolved by the cell. And this is on purpose so that it can, line up its chromosomes. You remember anaphase, telophase, all those things? Pull the little chromosomes apart? Yeah, the cell's getting ready to do that. This is what our filthy little papillomavirus has been waiting for. And so it doesn't even bother to get out of its vesicle. It's been hanging out in the trans-Golgi network for a while, and it's going to go over at this point. It's going to get dragged in on some sort of protein. We think maybe a kinesin, but not really sure. And it will actually make contact with the chromosomes. So we know the L2 will bind the L2 and the DNA to a chromosome. We don't know exactly what it's binding on the chromosome, if I'm reading the literature correctly. But yeah, now you have your L2 in a little vesicle on the chromosome. At some point now, L2 is going to break out that membrane disrupting part, which, if you remember from a couple slides ago, is down here, and it's loaded with basic amino acids. That's like a common strategy when you need to really disrupt membranes very fast. And so, yeah, it is going to now finally pop that bubble, and it'll just be L2 and the DNA running around. So L2 is going to act kind of like a nucleoprotein would. And L1 is going to act sort of like a spike protein would, except that there's no membrane there. It's just directly binding to the L2, which has the DNA on it. And L2 basically never leaves the DNA. It's always hanging around. All right. So now it's attached to a chromosome, and then after cell division, after we pull all those chromosomes apart, now of course we're going to reform two nuclei, and look who's in the nucleus now. Yeah, human papillomavirus, because it was attached to one of the chromosomes at mitosis. So again, it's probably the laziest way. Like, can't figure out a way to get into the nucleus or out of the nucleus. It's okay, just wait. Just lay there and wait. And eventually the nucleus will break down because you're in a basal cell. And that's what they do. They divide pretty rapidly. All right, so next step, how's the virus going to get copied? Well, you remember the virus doesn't copy itself. No, that's way too high effort for a human papillomavirus. It is going to bring in the E1 and E2 proteins, so those are going to be expressed because this is a little circle of DNA which is now attached to a chromosome in the nucleus. So yeah, genes are going to get expressed. Yeah, that's how biology happens, right? And the L1s are going to lay down here in the center. The L2s are going to sort of shelter around them, shepherd them into place. And then I think they eventually get done with the L2s, and the L1s are going to form this sort of thing. So it's two sets of 6 L1s. It's like a back-to-back hexamer, and they're going to be forming on the origin of replication of the virus. So this is a DNA virus. It's got to be copied by, as if it was a chromosome, you know, by the normal nuclear DNA polymerases. And so this is just a signal. It's a little recruiting stand. It's like one of those banners they put outside to recruit people to clubs. And it just says, protease, come land here. This would be a great spot to start a brand new origin of replication. And the whole holiday junction thing, yeah, all that stuff that you learned in Bio 1. So that's it. The virus doesn't do anything. It just makes an attractive little place for the cell to come in and do things. Yeah, again, that's very papilloma. Here is from a nice little paper where they actually show how the virus spreads. And it's going to spread at the mitosis phase. So as we are dividing the cell and pulling those chromosomes apart, there are little circles of DNA with L2 attached to one end and L2 reaching up and attaching to the chromatin at the other end. And that's enough. That attaches our viral genome to various chromosomes. It's not binding a particular place. It's probably binding to a protein that is already going to be bound to the chromatin, something like that. And so, yeah, once they are all attached to these, then as the chromosomes are retracted to the different sides, you get little papillomavirus genomes attached to those, sort of shaking them off. Yeah, isn't that adorable? Now, as it gets, as time goes on, it's going to start to make some L1 proteins, and the L1 proteins will eventually be able to grab these DNA circles and encase them in a little protein shell. And then when the cell finally bursts someday or gets, broken open by friction, that's when it's going to release a bunch of virions. But once again, L1 is not a high energy protein. It's not going to go into the nucleus to find these little guys and bring them out. As far as we understand right now, L1 probably waits until the nucleus is dissolved. Once again, yeah, ridiculous. And then it just packages these things up kind of lazily. And so now they're there. Yeah, some will stay attached to the chromatin, some will get packaged up. It's fine. Yeah, papilloma doesn't care. And that's basically how it's then going to spread. So there you go. That's the last thing that we covered. And that is the rest of the sort of mechanistic detail of how this virus does what it does. So, hope you found that useful. Thanks very much.